Learning outcomes. After studying this module, you shall be able to know about the sweat and the fecal matter, how we can examine the sweat stains, about the fecal matters, how to do the examination of fecal matters, importance of fecal matters examination. We will start with a brief introduction of sweat and fecal matter. Serology allows the forensic scientists to segregate these bodily fluids when found at the scene of the crime and then perform a variety of tests on them in order to identify where these fluids originated from, or most importantly, whom they came from. One important aspect of serology is determining whether or not stains resembling blood found at a crime scene are actually blood or some other stain that bears a similar resemblance. Sweat is a clear, salty liquid produced by glands of human skin, and it constitutes of water with tiny amounts of other chemicals like ammonia, dissolved salts and fatty acids. There exist numerous amounts of sweat glands in the body, and it is essential for thermoregulation. Like mammary glands, which originate from epidermal progenitors, the sweat comes out from the body through tiny holes called pores. When the sweat comes in contact of air, the air evaporates it and then the skin gets cool. Sweating is a normal phenomenon when environment temperature is high, or when we exercise, when we are anxious, or when we are ill. The rate of sweating increases. It also happens while being in a phase of menopause. If sweating is enormous, it is called as hyperhidrosis. Causes of hyperhidrosis include some health problems like thyroid or nervous system disorders, low blood sugar, etc. If the sweating is very little, then it is called as anhidrosis, and it can be life-threatening because our body may be overheated. Situation that causes anhidrosis may include dehydration, burns, and some skin and nerve disorders. Sweating is the excretion of fluid through the sweat glands of the body. The process of sweating is also called perspiration. Now we will study the module in detail with the help of visuals and graphics.
Now let us know how sweat are produced. There are three primary glands contribute to the production of sweat. These are the pseudoferous glands, that is, acrine and apocrine, and the sebaceous glands. The acrine gland is one of two types of pseudoferous gland in the body. These glands produce a secretion that is mostly water but contains many compounds in trace quantities. The average quantity of secretions produced during a typical 24-hour period varies between 700 and 900 grams. The pH of sweat has been reported to vary from 7.2, which is extracted directly from the gland, to 5.0 recovered from the skin surface at a low sweat rate, to between 6.5 and 7.0, which is recovered from the skin surface at a high sweat rate. The acrine gland also secretes organic compounds of primary importance to the development of latent print rich detail are the amino acids. The serine is the most abundant amino acid. The other pseudoferous gland present in the skin is the apocrine gland. These sweat glands are associated with the chorus hair of the armpit and pubic area. They are larger than acrine glands and secrete a thicker fluid. Now let us study The mechanism of sweating. Sweating allows the body to regulate its temperature. Sweating is controlled from a center in the perioptic and anterior region of the brain's hypothalamus, where thermosensitive neurons are located. The heat regulatory function of the hypothalamus is also affected by inputs from temperature receptors in the skin. High skin temperature reduces the hypothalamic set point for sweating and increases the gain of hypothalamic feedback system in response to vibrations in core temperature. Overall, however, the sweating response to a rise in a hypothalamic temperature is much larger than the response to the same increase in the average skin temperature. Sweating causes a decrease in core temperature through evaporating cooling at the skin's surface. As a high energy molecule evaporate from the skin, releasing energy absorbed from the body, the skin and the superficial vessel decreases in temperature. Cool venous blood then returns to the body's core and counteract rising core temperature. There are two situations in which the nerves will stimulate sweat glands, causing perspiration during physical heat and during emotional stress. In general, emotionally induced sweating is restricted to palms, soles, armpits, and sometimes the forehead, while physical heat induced sweating occurs throughout the body. People have an average of 2 to 4 million sweat glands, but how much sweat is released by each gland is determined by many factors, including the gender, genetics, environmental conditions, and age or fitness level. Two of the major contributors to the sweat rate are an individual's fitness level and the weight. If an individual weighs more, sweat rate is likely to increase because the body must exert more energy to function and there is more body mass to cool down. On the other hand, a fit person will start sweating earlier and easier. As someone becomes fit, the body becomes more efficient at regulating the body's temperature, and sweat glands adapt along with the body's other system. Sweat is not pure water. It always contains a small amount, that is 0.2 to 1% of solute. When a person moves from a cold climate to a hot climate, adaptive changes occurs in the sweating mechanism of the person. The volume of water lost in sweat daily is highly variable, ranging from 100 to 8000 milliliter per day. The solute loss can be as much as 350 millimole per day of sodium under the most extreme conditions. During average intensity exercise, sweat losses can average up to 2 liters of water per hour. Now, let us discuss the composition of sweat. Sweat contains mainly water. It also contains mineral, lactate, and urea. Mineral composition varies with the individual. Their acclimatization to heat, exercise, and sweating. The particular stress source, the duration of sweating, and the composition of minerals in the body. An indication of minerals contained is sodium of amount 0.9 gram per liter, potassium 0.2 gram per liter, calcium 0.015 gram per liter, and magnesium 0.0013 gram per liter. Also, many other trace elements are excreted in sweat. Again, an indication of their concentration is, although measurements can vary 15-fold, 0.4 milligram per liter, copper 0.3 to 0.8 milligram per liter, iron 1 milligram per liter, chromium 0.1 milligram per liter, nickel 0.05 milligram per liter, and lead 0.05 milligram per liter. Probably many other less abundant trace minerals leave the body through sweating. with correspondingly lower concentrations. Some exogenous organic compounds make their way into sweat as exemplified by an unidentified odoriferous maple syrup. Sentient compounds in several of the species in the mushroom genus Lactaris. In human, sweat is hypoosmotic relative to plasma, that is, less salty. Sweat typically is found at moderately acidic to neutral pH levels, typically between 4.5 and 7.0. Now, let us discuss the cause of excess sweating. Consumption of alcohols and caffeines. Patients of cancer during complex regional pain syndrome, emotional or stressful situations, essential hyperhidrosis, exercise, fever, infection, low blood sugar, known to be hypoglycemia, menopause, spicy foods, known as gastric sweating, warm temperature, withdrawal from alcohol or narcotic painkillers. Now we will proceed to the examination of sweat stains. Examination of sweat is very important from forensic point of view to analyze the blood group of any individual from their wearing apparels. The sweating bearing areas are first examined for the existence of urea by the Guy's urea nitrate test. If the positive result is found, then they are subjected to absorption inhibition, absorption illusion, or mixed agglutation test for identification of blood grouping antigens in them. Sweat from secretor individuals will contain A, B, and H antigen with regard to their blood groups respectively. Now we will learn about the Guy's urea nitrate test. Suspect stain is extracted with acetone, and after the acetone extracted is concentrated by evaporation. Filtrate the concentrated extract and then evaporate it to dryness. The residue is then reacted with the help of acetone solution and it is mixed through glass rod. Take one drop of solution onto a microscopic slide and then again it is permitted to evaporate. The urea crystal may be observed. The crystals are long colorless 4 or 6-sided rhombic crystals. Added nitric acid solution through glass rod, the urea nitrate crystals may be observed that indicates the presence of urea. Now let us discuss the forensic importance of a sweat. A latent print is formed as a result from the release of sweat. Principle behind this is that although uncontrollable variables such as the total amount of sweat deposited by the finger, the amino acid concentration of the individual's excretion, and the age of the fingerprint influence the amount of amino acids transferred to the paper. Amino acids are always present in perspiration in some amount. On contact with paper, these amino acids impregnate the surface of paper, where they are retained by their high affinity for cellulose. Some of these, because of this affinity, amino acids do not migrate significantly from their initial deposition sites. However, the amount of amino acids retained in the fingerprint decreases gradually over time. A latent print is a mixture of some or all of the secretions from the three types of glands. Latent print residues is generally divided into two basic categories, that is, water soluble and water insoluble. The water soluble portion of the print deposit is typically composed of acrine secretion like salt, example NaCl, and amino acids, example serine and glycine. Chemicals like anhydrine, which reacts with amino acids, and silver nitrate, sodium chloride, are effective reagents for visualizing this water-soluble portion of the residue. However, an acrine-rich latent print that is exposed to water most likely will not be recovered with these methods. That is why, before the introduction of physical developer in 1970s, there was no reliable method for recovering prints from water-soaked document. Latent print visualization with powder or dusting involves application of finely divided particles that physically adhere to the aqueous and oily components in latent print residue on non-porous surfaces. Latent print powder has an affinity for moisture and preferentially clings to the residue deposit by friction-rich skin. It is well accepted that the mechanical attraction between these particles and the moisture and oily components in a print causes adhesion, with absorption being a factor. Visualization will occur while reflected light, that is, light powders, absorbed light, that is, dark powder, and luminescence, that is, fluorescent powders. Commercial powder manufacturers tend to label powders by color, such as black, white, silver, gray, and so forth. Particles that serve as a good fingerprint powders include carbon black, that is colloidal carbon, lamp black, talc, aluminium, metal flake, and dolomite. Good binders include iron powder, lycopodium, cornstarch, rosin, and gum arabic. One of the most important and common latent powder known for its versatility and effectiveness is carbon black. Now we will start the second topic, that is fecal matters. Feces are a waste product from animal or human digestive tract expelled through the anus or colca by a process called defecation. Urine and feces together are called excreta. Feces are primarily a waste product of the human or animal bodily function. It nevertheless has several uses as fertilizer, soil conditioner, fuel source, and even medical purposes as a fecal transplant. In the case of human feces, during heme catabolism, waste product called bilirubin is produced. Bilirubin excreted in feces, which gives it brown color. Gut bacteria break down some of the bilirubin to urobilinogen, which is also excreted in feces. Bilirubin is a substance formed by the breakdown of red blood cells. It is passed from the body in stool. Bilirubin is not found in urine. If it is present, it often means that the liver is damaged or that the flow of bile from the gallbladder is blocked. Now, let us have an idea about the physical appearance of feces. Stain appearance characterized by green-brown color caused by bile pigments and their breakdown products. Odor caused by breakdown of amino acid into indole, scatol. and methyl mercaptan by bacteria. It can also be identified by the presence of microscopic components of foodstuff by using Lugol's iodine. Now let us discuss the examination of fecal matters. The identification of fecal matter is very important, especially in case of sodomy. The following examinations are carried out in order to identify fecal stains. First one is microscopic examination. Second is chemical examination for the detection of bilirubin. And third one is DNA typing of feces. Firstly, we will discuss microscopic examination in detail. In the microscopic examination, the suspected stained area is softened by the use of normal saline for half an hour. A small amount of scrubbing from the stain is taken on a microscopic slide, and 1 or 2 drops of normal saline and a drop of Lugol's iodine is added to it. The microscopic slide is covered with the cover slip and examined under the microscope for the determination of undigested food particles like vegetables residues and muscle fibers. Sometimes the cells like pus cells, epithelial cells, cysts of entoamoeba histolytica, and gradia intestinal ova, intestinal helminthes are observed along with large numbers of bacteria. Now, secondly, we will discuss the chemical examination for the determination of bilirubin. A portion of suspected stain is extracted with a little amount of distilled water and added 2 to 3 drops of 10% of mercury chloride solution, and amyl alcohol is added as equal to that of water to the mixture, and the whole mixture is shaken thoroughly. The supernatant alcohol layer is pipetted off in another tube, and then 2-3 drops of 10% alcoholic zinc chloride solution is added to it. Under UV light, rose pink coloration of with beautiful green fluorescence is observed that indicates the presence of bilirubin. This test is considered to be one of the very sensitive and reliable tests. This test relies on the formation of a green fluorescent zinc urobilin complex formed in the presence of neutral alcohol zinc salt. Example, alcoholic zinc acetate. Thirdly, we will discuss about the DNA typing of feces. About 1010 cells are lost from GI tract per day, but nuclear DNA testing is unsuccessful in feces because of the inhibitory effect of bile pigments even at low concentration, and the large amount of bacteria and digestive enzymes also serves to degrade the DNA. Mitochondrial DNA testing has proven more successful with these kinds of samples. Now let us know the forensic importance of fecal matter examination. Fecal matter as evidence is always encountered in cases like sodomy and bestiality. If the fecal matter is already present in the crime scene, then there is a possibility of transfer of fecal matter through the footwear or even on the garments of the suspect. In sodomy and bestiality, fecal matter as evidence may be found on penile swab as well as in the garments. Fecal sample tissue paper smeared with fecal material and clothing that contains human feces may be the only evidence left at a crime scene. Thus, human stool may be a valuable source for forensic DNA evidence. However, due to microbial presence in the gastrointestinal tract, the extraction of human DNA from feces may be challenging. Also, stool contains high levels of compounds that can degrade DNA and inhibit downstream PCR reactions. These compounds must be removed for reliable analysis of extracted DNA. Fecal matter plays a vital role in examination of drug dependency nature of the suspect. It has also been observed that the color of fecal matter becomes green, black, and red due to consumption of various drugs. It can also be used for the purpose of grouping of blood and hence it can help in person's individualization.
Now let us summarize what we have learned in this module. Sweat is a clear, salty liquid produced by glands of our skin, and it is made of almost completely of water with small amounts of other dissolved chemicals like ammonia. Sweating a lot is normal when it is hot or when we exercise, are anxious, or have a fever. It also happens during menopause. If sweating is too much, it is called as hyperhidrosis. If the sweating is very little, then it is known as anhidrosis. It can be life threatening in antidotes because our body may be overheated. Sweating is the release of liquid from the body's sweat glands. This liquid contains salt. This process is also called perspiration. The sweat bearing areas are first examined for the occurrence of urea by the GE's urea nitrate test. Feces are a waste product from an animal or human digestive tract excreted from the anus or cloaca during a process called defecation. Under UV light, rose pink coloration of the mixture with beautiful green fluorescence is observed, which indicates the process of bilirubin. Generally, bilirubin is not present in urine. If it is found, then it is due to the damage in the liver or that the flow of bile from the gallbladder is blocked. A positive test result of the fecal examination shows gastrointestinal parasitic disease.